Northern blotting is a technique used to study RNA expression by detecting specific RNA molecules within a mixture of RNA. The procedure begins with the extraction of total RNA from cells or tissues. The RNA is denatured using chemicals such as formaldehyde or glyoxal, which stabilize it in a single-stranded form as RNA molecules may naturally fold back on themselves through internal base pairing, forming secondary structures. Denaturation is important because it prevents RNA secondary structures from affecting migration during electrophoresis. It also ensures that the probe, which is a single-stranded nucleic acid, can later bind accurately to the complementary RNA sequences during hybridization. The denatured RNA samples are then separated using denaturing agarose gel electrophoresis, which maintains the RNA in its single-stranded state throughout the separation process. The RNA molecules are separated mainly according to size. Smaller RNA molecules move faster through the gel, while larger molecules move more slowly. For a more detailed explanation of electrophoresis, you may refer to the related video. After electrophoresis, the gel may be stained and visualized to confirm that RNA is present, intact, and has separated correctly. The migration distances of the RNA ladder bands can be recorded and later used to estimate transcript size by comparison with RNA size markers. However, at this stage, only the total RNA population is visible, and the specific RNA transcript of interest cannot yet be distinguished from the many other RNA molecules present in the sample. Identification of the target transcript is achieved later using a labeled probe that hybridizes specifically to the RNA sequence of interest. The separated RNA molecules are then transferred from the gel onto a positively charged nylon membrane through capillary transfer. This transfer step is necessary because agarose gels are fragile and do not allow efficient probe hybridization, repeated washing, or long-term preservation of the RNA pattern. In contrast, the membrane provides a durable surface where the RNA can be immobilized and probed more effectively. During capillary transfer, blotting paper soaked in transfer buffer draws liquid upward by capillary action, carrying the RNA molecules from the gel onto the membrane. The membrane is carefully placed on the gel without air bubbles, followed by layers of filter paper and a weight to ensure close contact. Once transfer is complete, the membrane contains an exact copy of the RNA band pattern from the gel, with the RNA molecules now on a durable surface for subsequent detection. But first, the RNA must be immobilized on the membrane because it is initially attached only weakly. Immobilization is achieved either by exposing the membrane to ultraviolet light or by heating it to around 80 degrees Celsius. This fixes the RNA onto the membrane and prevents it from washing away during later steps. Before adding the probe, the membrane undergoes a pre-hybridization or blocking step. Blocking solutions containing substances such as Denhart solution or salmon sperm DNA coat empty binding sites on the membrane. This prevents non-specific binding of the labeled probe and helps reduce background signals. Next, the membrane is incubated with a labeled single-stranded nucleic acid probe. During hybridization, the probe binds specifically to complementary RNA molecules on the membrane. Unbound probes are then washed off. Traditionally, radioactive isotopes were used to label probes, and detection was carried out by exposing the membrane to X-ray film, producing dark bands where hybridization occurred. Today, non-radioactive methods are more commonly used because they are safer and easier to handle. For example, fluorescently labeled probes can be detected using fluorescence imaging systems to observe the RNA transcript if present. Pause here and try the quiz question on the northern blot procedure. Now let's look at some advantages of northern blotting. Unlike RT-PCR, which mainly measures RNA abundance, and conventional RT-PCR, which mainly detects specific RNA sequences, northern blotting also provides information about transcript size together with relative abundance. Transcript size is determined from the position of the RNA band compared with RNA size markers, while relative abundance can be estimated semi-quantitatively from band intensity. This allows researchers to identify alternatively spliced transcripts or degraded RNA molecules. Although newer techniques such as RNA sequencing are more sensitive and high throughput, northern blotting remains a valuable method for confirming RNA expression and transcript size. Pause here and try another question on northern blotting. If you found this video useful, do check out the other videos on the channel and subscribe so you do not miss future content. Also remember to like the video and support the channel.